So Ribisco, or ribulose 1,5-biphosphate carboxylate oxygenase, is the enzyme that catalyzes the carbon fixation process, but as the name implies, it also will bind with oxygen. So here is the active site. So we can either have carbon dioxide come and enter that carbon dioxide and then go through the Calvin cycle as usual, or that as a competitive inhibitor, oxygen can also fit into that active site. Right? Plants will choose to close their stomata to prevent water loss, which means that they're no longer being able to access carbon dioxide. The plants will go through the Calvin cycle until their carbon dioxide is used up. That's going to lower the concentration of carbon dioxide and increase the concentration of oxygen. So that means that more likely oxygen is going to be entering the active site of Ribisco. But what's going to happen if Ribisco binds with oxygen? Well, what will happen is this process of photorespiration. So we're seeing oxygen coming to the active site of Ribisco. We don't have enough carbon molecules in order to build our glucose molecules. So what we have to do then is go through this wasteful process where we're using ATP and we're not producing glucose in order to get our UBP back so that we can bind with carbon dioxide. So this means that we're spending energy and not getting sugar, so plants are not going to grow as well. And in places where it's often hot and dry, we've seen the evolution of two alternative photosynthetic pathways to prevent photorespiration. In C4 pathway, Ribisco and oxygen are kept separate in space. We see this special anatomy, we call it Kranz anatomy. Kranz means wreath in German, and so we have this wreath-like these cells that surround the vascular tissue. And what will happen is carbon dioxide will enter the leaf, be picked up by this special enzyme called PEP carboxylase. And this one carbon dioxide molecule, right, we've got one carbon in there, is going to bind with the 3 carbons in our PEP molecule that we see down here. And that is going to give us a 4-carbon malate sugar molecule. And then that malate then is transferred into this bundle sheath cells where malate is broken back down into carbon dioxide and into PEP. And we're keeping, and oxygen's not allowed to be present, right? So in these bundle sheath cells, once that malate is broken back down into PEP and carbon dioxide, then the Calvin cycle can pick up that carbon dioxide and go through the process as usual. In CAM photosynthetic pathways, we keep oxygen and Ribisco separate in time. So the CAM photosynthetic pathway stands for crassulacean acid metabolism. It's named after the plant family, the Crassulaceae family, where this pathway was discovered. In this pathway, stomata are only open at night. So during the daytime, no carbon dioxide is entering the plant. And they do this in order to prevent water loss. But hold on a second. We paid attention earlier, and we know that photosynthesis cannot occur without having light energy. So how are we going to do photosynthesis at nighttime? Well, what happens is at night when their stomata are open, carbon dioxide is going to join with PEP using that PEP carboxylase and convert into that 4-carbon sugar molecule called malate. In the daytime, when there is light, malate is going to be broken back down, releasing carbon dioxide, and then we're going to have the Calvin cycle continue just like we would in a C3 plant. Now, unlike the C4, we don't have to have any internal specialized anatomy. And so most CAM plants will actually switch back and forth between CAM and C3 depending on those environmental conditions. Except for our friend the saguaro. The saguaro can only do CAM photosynthesis. I asked my friend who is one of the world famous researchers of saguaro physiology, and I asked him why can't saguaros do C3 photosynthesis? Most plants can switch back and forth, but why not saguaros? And he gave the very scientific answer of, I don't know. The regular way to do photosynthesis is the C3 photosynthetic pathway. The C4 photosynthetic pathway keeps rubisco and oxygen separate in space. We have a specialized anatomy that allows us to do this. We're going to use the 4-carbon sugar malate, and that's where we get the name from, 4 carbons in malate. The CAM photosynthetic pathway keeps rubisco and oxygen separate in time, and we don't have any specialized anatomy for this, so most plants can switch between CAM and C3.